Hi everyone. So yes, this is me. Wait, I'm gonna find my presentation. Um, okay. One second. Okay. So can you hear me? Great. So I'm going to talk about a paper we published 2 years ago. Um, we tried to take the world of BCI outside of the outside world, actually, and we tried and succeeded of implementing a method of using the algorithm result in brain-computer interfaces to controlling some aspect inside a living organism. So, as we all know, current BCI is dedicated and focused on motor control, rehabilitation, communication, and mostly assistance for impaired individuals and some for games. What we try to suggest is a novel method of using BCI or neurofeedback for active temporal control of drug delivery. Why is this important? So we have a few methods of drug delivery that are well known and used. We have a controlled release drug, such as you take a pill and it's encapsulated in a substance that makes it release slow. We have a quick release, slow release, and we can control the amount of time it takes for a substance to be fully released in the body. What we can't control is when we want to start and stop the effect of that substance. For example, an individual with ADHD does not need that amphetamine to be active in his body during of the whole day. He wants it to be effective during an exam, during a meeting, or just during something that he needs that drug to be effective in. But current technology only provides him with methods of taking the drug and then being affected by the drug between 6 hours to 24 hours, which is very, very limiting, and it causes a lot of negative effects on the usability of the drug and the overall effect of using that kind of drug. So, for this purpose, we decided to take BCI technology and algorithms and combine it with a different technology called DNA origami, which is a derivative of DNA nanotechnology, and try to control the DNA origami inside a living organism to release a specific drug when we want it to be released. In 1991, Ned Seaman developed a method called DNA nanotechnology that relies on naturally occurrent holiday junctions within the mitosis operation of a dividing cell to construct structures out of a single-strand DNA. He devised a method to calculate which strands can be joined together, and then he managed to find a way to create a cube, a nano-sized cube, only out of DNA strands. So this is how it looks like. You have 4 specific DNA strands, which complement only half of the other DNA strand, which then result in a 4-dimensional holiday junction, and which can be then folded into a 3D cube or 3D object of your choice. In 2006, his PhD student developed a way to enhance the ability of creating these structures by using a viral plasmid, which made the possibilities basically endless. He, instead of relying only on short DNA strands, which are limited by their size, we now can take a long viral DNA strand, which is comprised out of approximately 2,600 bases of DNA, and then we fold those to bigger resolution structures. Bigger, I mean, instead not being 7 nano, but being around 50 to 100 nano, being 30 nano, still small, but more functional. On these structures, we can imply and use specific locations that are functional for activation and interaction in the organism. This is how it looks like. This is the basic operation. This is a real image of a DNA viral strand. It's considered to be a template, and then we add shorter strands, which are named staple strands, that reduce the possibilities of the main strand, and they fold it, quotation marks, fold it to a structure of our choice. And this is how it looks like at the end. Okay. So, in 2012, in George Church's lab in Harvard, my PhD supervisor developed a method that takes this basic concept, utilizes two structures, and join them together to be what later was named a DNA nanorobot. This DNA nanorobot has possibilities of carrying specific proteins, which are drugs in the end, and locking them inside a mechanism that can be controlled from the outside. We wanted to use this method to see if we can control the release and the activation of the drug inside the user. We developed a very, very simple algorithm that calculates basically the amount of working load in a participant. We have a no working load and then a high working load, a very binary algorithm that decides if the participant is in a situation where he needs this drug assistance or not. We used an insect that was used previously in testing of this kind of technology. The insect was inserted with the DNA origami that withheld within it a fluorescent proteins that we can then measure the effect of. And then we recorded EEG signals from participants, created a classifier, which then led to a RF-MF generator. We then surrounded the insect in an aluminum coil and then heated up the origami that was locked using iron oxide, which are nanoparticles that can release the coupling in a specific temperature. We initially analyzed the effectiveness of this method, and we saw that after heating of 3.2 minutes, the fluorescence rises, meaning that the nanorobot's open. This is in a vial, not in the insect itself. And then we also measured to see the sustainability of this technology, this method. I have to say that you have to understand that the origami itself is a capsule are very, very, very stable. They last for years and years in room temp, and It's a viable technology to send and use around the world. Later, we did the same within the insects, and we crossed the activation of the coil to the working load, meaning that when the participant was confronted with difficult arithmetic problems, he showed a working load in the classifier, and then we extracted some plasma from the insects and measured to see which insects were affected by the opening of the origami or not. So here you see that when the DNA gated robots, meaning they can't open using the nanoparticles, show low fluorescence, and compared to the NP, which is nanoparticle gated robots, together with the cognitive load or open robots, show the same fluorescence. This means that the DNA origami that was inserted into the cells opened up when we induced it using the field generator, and then they stained the cells within the insect, and then we controlled the feedback of this system using an EEG cap on a individual. This was a proof of concept of showing maybe a new method of using the algorithm we all develop in the BCI world to maybe have a new and different kind of interaction, not outside the world, but inside our bodies. I'm sure you can think of many methods that this technology can be used. It's not FDA approved yet, but we're hoping to get there eventually. This was work done by a few of computer students, me and a few students from the Bachelet lab at Bar-Ilan University, and advised by Doron, Yaakov, and Ido Bachelet from Bar-Ilan. Thank you.